Welcome to AP Biology. Today I want to go over restriction enzymes and gel electrophoresis, which is a little ahead of where we are, but you'll need to know this for the lab. And then once we get to the biotechnology unit, you'll already know a whole bunch of stuff. So restriction enzymes are enzymes, so they speed up chemical reactions. They cut DNA. That's what they do. They're actually originally from bacteria, and it was a sort of a way to protect themselves from viruses. So the term restriction actually came from the fact that bacteria could protect themselves from just a restricted number of viruses, and the viruses they would be protected from would be the ones that they could recognize and cut. And so this particular restriction enzyme cuts right here. And there are a bunch of different restriction enzymes, and each cuts at a different particular sequence of DNA. So first I need to go over some definitions. A plasmid, this is a small circular DNA molecule that replicates separately from a bacterial chromosome. So this is a bacterial cell. That made it clearer, right? Because you can see the word bacterial cell there. Whoa. Anyway, here's the chromosome, which is the DNA that that generally has most of the genes that the cell needs to live. These plasmids, though, these are extra chromosomal, meaning not part of the chromosome. And bacteria can actually swap these, and they might have genes for particular other traits that might help the bacterium to live. Okay, so a restriction enzyme is an enzyme produced by certain bacteria that cuts DNA molecules at specific sequences of bases. So E. coli has a couple of restriction enzymes, and Streptococcus has other restriction enzymes, and we use those in the lab. So for example, the Hint3 digest cuts every time this enzyme can actually recognize a segment of DNA. So every time this particular enzyme sees A, G, C, T, T, it will cut it between the A's right here and right here. And it'll leave what we call sticky ends, which means that there are some bases here that don't have a partner and some bases over here that don't have a partner. And those sticky ends are going to actually be important when we come back to biotechnology and talk about it later. Here's another different restriction enzyme that happens to cut every time it sees C, T, G, C, A, G. Notice the other direction, right? Look at the other direction of DNA. It goes C, T, G, C, A, G. And we haven't talked about how DNA is anti-parallel very much. I think we mentioned it very briefly, but we'll come back to it later with the DNA unit. But for now, just know that DNA runs sort of in opposite directions on each side. And so this is going 5 to 3, and this is going 5 to 3, right, in the opposite direction. And notice that the letters are the same if you read them from 5 to 3, CTG, CAG. Okay, so this one cuts between the G and the A, and over here, also between the G and the A. And so that's also leaving these sticky ends or protruding ends. And then this final one down at the bottom is cutting a little bit differently. So whenever this particular enzyme, the EchoRV, whenever this particular enzyme sees GATATC, oh, and this way it would be GATATC, right? Same code, just running upside down to each other. Whenever it sees that, it cuts between the T and the A, and this time it leaves these just flat ends, not sticky ends, these blunt ends. Okay, so restriction enzymes often cut at palindromes. So a palindrome is anything that reads the same forwards and backwards, and in English it might be something like race car, or I don't know, what are some others? Taco cat, I think, is another one. Anyway, they're the same when read 5 to 3' on one strand, and then also 5 to 3' on the other strand, on the complementary strand. Remember, this is 5 to 3 in one direction, which means it would be upside down on the other strand, right? So here's my palindrome for you. I'll impress her with a palindrome. Madam, I'm Adam. And she comes back with Eve. It's kind of cute, right? There's your mom joke for the day. All right, so the most useful restriction enzymes cut in a staggered way, producing these sticky ends right here. And that's actually not important for the unit that we're on right now, but it will be important when we come back and talk about biotechnology in a few months. Okay, so scientists can use these enzymes. They're made by bacteria, but we can use them in the lab. And that's really the key to almost all of biotechnology. It's something that exists in nature because nature's had lots and lots and lots of times to lots of time to make these, to evolve these really cool uh, enzymes or other things that we use in the lab as tools. So scientists take these enzymes from bacteria and we can use them in the lab for various purposes. Today we'll talk about one of those purposes. So one of them is to cut DNA and then pull the DNA through a gel in order to see the different fragments that are left behind by the restriction enzyme. So now we're going to talk about analysis of DNA fragments using gel electrophoresis. So let's get me out of the way if possible here. There we go. Okay, so DNA from different sources can be cut by the same restriction enzyme. So let's say we have where's my pen? Okay, good. Sorry, let's say we have some normal DNA and then some mutated DNA. So here's the normal DNA. And there's some particular restriction enzyme, MTS1, maybe? I don't know if that's an L or 1. But anyway, it happens when it sees normal DNA. It cuts it here and here and here because the sequence of DNA that this particular enzyme recognizes happens to show up 3 times. So if you have a mutated piece of DNA, so here's the mutated one, it just so happens that the A's, T's, C's, and G's right here at the restriction site, the place where this particular restriction enzyme recognizes, the mutation messed it up. And so now that particular A, C, C, G, whatever it was, isn't the same anymore. So now that's not being cut the same as it was before. So you can cut them with an enzyme and then pull them, you can separate them with gel electrophoresis. So here is the restriction enzyme cutting the DNA here and here and here, and this one's cutting here and here. Notice that with this top one, you've got a couple of fragments here, plus however many other fragments are downstream and upstream. But this one, because this restriction site is missing, instead of getting 2 different fragments here, it's got one bigger fragment. So you can take those samples and you can't see them. DNA is way too little and clear. You just can't see it. But if you replicate it a bunch of times and then you, which we'll talk about later, and then you expose it to these restriction enzymes and then make it show up with a dye, and then pull it apart with gel electrophoresis, you can see different fragments. So this is a fragment of DNA and that's a fragment of DNA, and notice how they're the same. Ooh, until you get to this one, and that's different. So there's your difference, and then here's another difference, and let's look all the way down. Looks like we've got another one here. So do you see how the fragments are all the same? So you've got a long chain of DNA, and you've copied it a bunch of times for the purpose of this lab, and then you cut it with a restriction enzyme, and you're left with a whole bunch of fragments, little pieces of DNA. And the normal one has these 2, whereas plus a bunch of others. And the mutated one just has one, let's say just one restriction site messed up, one place where the enzyme is supposed to cut that it can't cut anymore. So now you have a big fragment instead of 2 little ones. And so here we see, let's change colors here. Here we see the differences right here. This is a bigger fragment, which I'll show you in a minute. And then the normal one has 2 littler fragments. So let's look at the size of these. So mutations can change the number of restriction sites for a particular restriction enzyme. We just said that. This will produce a different number of fragments and also change the sizes of the fragments. So instead of having 2 little fragments up here, now we have one bigger fragment. And so how does that get pulled down, get pulled through the gel? And how do we know how big the different fragments are compared to each other? We'll talk about that in just a minute. I have one other thing that I want to show you that sometimes will make the fragment sizes different besides a mutation of this type. So here's another example of a mutation that could happen. So let's say you have somebody's DNA here and there are 24 repeats of GT. It goes GT, 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 and on the other side, TG, TG, TG. Sometimes little repeats like that will be copied, and so this person has a whole bunch of extras of those GTs in a row, and this person, maybe it's the same person, does too. And so you might get different fragments of DNA because there could be a restriction site here, and that would actually be here and here. And so this person's DNA is going to be that fragment will be a little bit bigger, not because there was a mutation at the restriction site, but because they just have a bunch of repeats that the other one doesn't have. Okay, so we'll talk about how gel electrophoresis works in a minute, but first I just want to talk about why you would use these restriction enzymes. So there are many reasons to expose DNA to restriction enzymes in the lab. One of them is called DNA fingerprinting, and it includes forensics and paternity tests. So for example, with this bottom picture, here's a victim, here's DNA that was found at the crime scene that's not the victim's, and you can see just the fragments of DNA get pulled through differently and look different. Here's suspect 1, 2, and 3, and you can see that suspect 2 matches the DNA at the crime scene. And then paternity test, very similar type of an experiment. And so what's happening here is here's the mom and here's the kid. And so the kid shares some fragments in common with mom, right? Like these, but had to have gotten the others from like their other chromosome, right? From dad. And so it looks like these two dads look like they match this one, or possible dads, I guess. And it looks like these two match, match this one. So dad number 3 is probably the child's dad. Okay, so here's a scientist loading DNA samples into a gel. Each of my cohorts who come in in person, I hope will get a chance to load those gels. For those of you who are remote, I'm going to show you a video of that. Okay, so here's what actually happens. Here's the gel right here. You can see it kind of ending right here. It's in a little plastic container, and then you load these wells. They're not, if you look at them from the side, they're not all the way through. They're these little wells like this. And so here's, let's say here's the rest of the gel, right? So it's got a certain thickness to it, and the wells sort of maybe look like that, right? So they're these little, so here's the well where you're going to put the DNA, but the rest of it is actually like an actual like gel right at the top here. So the holes don't go all the way through, right? The very bottom of the gel is here. So you want to put your sample into this space right in here. So it's a little depression. It's not a hole all the way through to the bottom, but it's a little depression, and it's made by sample combs. So okay, so you load your samples into the gel. Maybe this is the DNA from a crime scene or something, and this is DNA from different suspects. Or what we're doing today with, it's going to be, or what we're doing in lab is going to be viral DNA that we cut with different restriction enzymes. Okay, so then your DNA is negatively charged. And so what you do is you put the negative electrodes here, the black ones are negative and the red ones are positive, and you line it up like this because if DNA is negative, it's going to be pulled toward, I don't know if you can see that, it's going to be pulled toward the positive electrodes. And so this thing is a power source, and here's the gel. This thing has ions in it. It's just some liquid with some water with some ions, electrolytes in it. And you fill the liquid up over the gel, and then you plug it in, put a cap on it, and pull those samples through. Whoops. And so the gel matrix is made out of this material that's sort of like an obstacle course. The big fragments can't go as fast. So picture some little DNA molecules sort of running toward the positive side. The small fragments go further, and the big fragments have more, it's tougher for them to get through all of the materials of the gel, so they go more slowly. So smaller fragments of DNA will move more quickly through the gel matrix. So you'll usually use a standard that you buy from the lab. Sometimes you call it a ladder, but usually you call it a standard. And so they'll sell it and they'll say, hey, this standard has a sample piece of DNA that's 100 base pairs. And we have another one that we put into this sample that has 200 base pairs, 300, 400, and 500 base pairs. Because if you put your samples in the wells up here and you run the gel for 10 minutes, the fragments are only going to go so far. But if you run it for 30 minutes, they're going to go further. So you're going to run it for however long you run it, and then you can, you'll put it, like I'll show you how later, but you'll make these show up with a dye. So anyway, if you run it for 30 minutes, maybe the one that's 100 base pairs goes to here. That way, if you're running your standard plus your samples and you don't know how big the fragments are for your samples, that way you can tell how big the fragments are for your samples. So like this one, for example, is I think between 100 and 200 base pairs, whereas this one is between 300 and 400 base pairs. So gel electrophoresis separates DNA. You can actually separate other molecules too, anything that has a charge to it, like RNA or proteins can also be separated that way. But in the lab, we're going to separate DNA molecules by fragment size. The little ones go through further. So notice the wells right here. So these must have been running that way. So that would be a littler fragment and that would be a bigger fragment. Okay, so here's just another example of why you would do gel electrophoresis. Restriction fragment analysis can be used to compare two different DNA molecules, such as two alleles for a gene, if the nucleotide difference alters a restriction site. So this is an example that's very much like one I saw on an AP test at one point. So this is the beta globin gene, which is from hemoglobin, and here's someone who has normal, this is the normal allele, which means alleles you have like one from mom and one from dad, right? And here might be the normal one, and here's the mutated one or the different one. So D-day1, I think, is some restriction enzyme from some bacterium, and it happens to cut here and here and here and here. So you're not using the whole, all the 46 chromosomes from an entire human. All you're using is that one gene. So we'll talk about a tool later called PCR where you can just copy one particular gene if you want to. But I want you to imagine that you've copied this beta globin gene from someone who has the normal allele, and you've copied it a gazillion times. So you have one long piece of DNA, and then you expose it to the D-day1 gene and you cut it here and here and here and here. And so what you've got is 1, 2, 3 fragments. And then let's say you also copy this DNA for someone who has sickle cell anemia, and so they have a different or a mutated B globin allele. Everyone has lots of mutations, right? But so this is just a different one from the wild type, the one that most people have. And that restriction site right here, the one that used to be right here, that's where the mutation is for sickle cell anemia. So when you copy this DNA a gazillion times using that technique we'll talk about later, and then you expose it to this particular restriction enzyme, it will cut it here and here and here, just like the regular person's, but it's missing that restriction site. So now instead of 1, 2, 3 fragments, now you only have 1, 2 fragments. And so when you put that into the well, these are not pieces of DNA, that's just the well, the little depression that you put the DNA into. The big fragment here is the same. It didn't go very far, right? So here are the bigger fragments. But these little fragments, right, this one has sort of a medium-sized fragment, and so it ends up here. But that fragment got cut in half on the normal person's DNA right here. And so that fragment is in 2 pieces over here for the normal person. If you had someone who had DNA and they were heterozygous, if you remember what that means. Let's say you have someone who's heterozygous, like, and you put their DNA into a well right here. Let's pretend that the gel was much bigger, right? And you put in both the big N and the little n DNA into this well, you would get this normal, this big fragment. So you'd get the DNA here. And then from this allele, you'd get this medium-sized fragment, which would be here. And then you'd get these 2 littler ones, which would be here and here. So this person had big N, the normal DNA from both parents. This one had the sickle cell anemia uh, fragment from both parents, and this person was heterozygous, so they had all of the bands. So kind of cool, right? All right, so variations in DNA sequences are called polymorphisms. So poly means many, right? Meaning at least more than one. Morph is your shape, which in terms of DNA is your letters. It's the A, T, T, C, G, whatever your letters of DNA are, whatever the nucleotide sequence is. And so if you have polymorphisms, that means you have different variations for a particular sequence. So let's say you have a polymorphism. This person is different from this person for their DNA for this particular gene. So it's polymorphic. They're different for their order of nucleotides. restriction site is the place where a particular restriction enzyme will cut. And so that's different, right? So restriction fragment length, what the heck does that mean? Well, if you use a restriction enzyme to cut the DNA, that particular piece of DNA, you're left with this fragment, this little piece of DNA, and that fragment, and that fragment. But if you use that same restriction enzyme on this mutated DNA, this different DNA, you have different restriction sites, right? Because one is missing. The one that was right here is missing. So you're only cutting it 3 times. And so you've got this one and this one. So we have two different, so we have different fragment lengths, right? We have this one compared to these two. And maybe you're saying what happens to the DNA upstream and downstream. Maybe that's part of like either they didn't copy it, right? Or it's like such a tiny one, it went through really fast because maybe they only copied to right here. So all we're looking at, don't worry about the ones on the ends, all we're looking at are these particular ones. Okay. So RFLP is the short name for it, but it means restriction fragment length polymorphism, meaning there are different restriction sites because this particular enzyme happens to cut at a place that got mutated right here. And it ends up giving you different lengths of DNA, different fragments of DNA. All right, that's the story.